Hello everyone and welcome to Gurukul classes. In this today's lecture, we are going to see some of the general characteristics of Archegoniates. We will continue from our previous discussion and we will move on to the evolution of certain features in land plants. No discussion of evolution of plants on land can be undertaken without a brief review of the timeline of the geological eras. So, the geological timeline, this era, an early era known as Paleozoic, is divided into 6 periods. It starts with the Cambrian period, followed by Ordovician, Silurian, Devonian, Carboniferous and Permian periods. More than 500 million years ago, the major event to mark the Ordovician was the colonization of land by the ancestors of modern land plants. Land plants first appeared during Ordovician period. The evolution of plants occurred by a gradual development of novel structures and reproduction mechanisms. It occurred by a stepwise development of physical structures and reproductive mechanisms such as vascular tissues, seed production and flowering. So, let us understand how these structures gradually got evolved. In this diagram, if you could see, as we move from charophytes towards embryophytes, first non-vascular plants got developed and then the vascular plants got originated. The evolution of seed was also gradual. Initially, it was the seedless plants and then the seed plants gradually started developing, which is represented by gymnosperms and angiosperms. So, let us talk about the origin of vascular tissues. The emergence of the tracheophyte based vascular system of land plants has major impacts on the evolution of terrestrial biology in general. It facilitated the development of plants with increased stature, photosynthetic output and ability to colonize a greatly expanded range of environmental habitats. In combination with prevailing global climate change, which included dramatic changes in atmospheric carbon dioxide levels, gave rise to the development of the cuticle and stomata. These important adaptations reduced tissue dehydration and increased the capacity for exchange of carbon dioxide, thereby enhancing the rates of photosynthesis. Following acquisition of these two traits, early land plants evolved cells specialized for long distance transport of food and water. Irrespective of plant group, these cells became arranged end to end in longitudinal files having a simplified cytoplasm and modified end walls designed to increase their intra and intercellular conductivities respectively. The two essential functions that are performed by the vascular systems are delivery of resources such as water, essential mineral nutrients, sugars and amino acids to the various plant organs, and the provision of mechanical support. The development of vascular tissue by land plants is generally recognized as one of the most important evolutionary steps in the colonization of terrestrial habitats. Both bryophytes and tracheophytes have developed conducting tissues consisting of cells specialized for the distribution of food and water throughout the plant body. In land plants, the degree of cellular modifications of transport cells increases from the bryophyte, that is pre-tracheophytes. They are also termed as non-vascular plants represented by liverworts, mosses and hornworts, to the early tracheophytes, that is the vascular cryptogams, which is represented by lycophytes and pteridophytes, and on through to seed plants. Studies based on fossil records and extant bryophytes have established that developmental programs evolved to form specialized water and nutrient conducting tissues. Based on the fossil record, early pre-tracheophyte land plants appear to have developed simple water conducting conduits having smooth wall with small pores, likely derived from the presence of plasmodesmata. Similar structures are present, for example, in some of the mosses, the most ancient being termed as water conducting cells, that is WCC, and the more advanced being the hydroids of the peristomate mosses. In this diagram, which is a transverse section of a leaf of one of the mosses, we can very well see that in the center there is the presence of sterites, some of the mechanical tissues, and followed by hydroids, and these hydroids are surrounded by a larger cells also called as leptoids. We will see that these leptoids have gradually later on evolved into phloem tissues, and the hydroids have evolved into xylem tissues. The plant vasculature is a sophisticated system that has greatly contributed to the evolution of land plants over the past few 100 million years. The formation of vascular system is a well organized plant developmental process. The remarkable event of evolution from the bryophytes to pteridophytes was the evolution of dominant and independent sporophyte phase, which symbolized the true root, stem, leaves and vascular tissues. Present day plants with leaves and branches are the result of this continuous evolution in the axial sporophytes of the primitive land plants. Earliest vascular land plants such as Rhynia, Horneophyton, Xylophyton, Asteroxylon had a simple dichotomous branch axis and bore sporangia terminally or laterally. The developmental trends in the conducting tissues of mosses elucidate that bryophytic conducting systems can contribute to defining the structural level of conducting tissues in the ancestral types of land plant that gave rise to bryophytes and tracheophytes. The conducting tissues of mosses begin with phloem-like cells, the leptoids, and the xylem-like cells, the hydroids. In this diagram, the hydroids are small cells which are surrounded by leptoid cells which will gradually later on develop xylem cells. Now let us understand what are leptoids. Leptoid is a cell type found in both phases of some mosses and resembles the primitive sieve element of vascular cryptogams and particularly ferns. Because of this similarity with vascular plant sieve elements, Benke in 1975 suggested the use of the term sieve element of bryophytes to describe the leptoids. As in tracheophytes, the food and water conducting tissues of mosses are specially related. In a transverse section of gametophore, the food conducting system known as leptome is located as a sheath of cells which surrounds the central cylinder of water conducting cells. The leptome consists of leptoids and associated parenchyma, whereas hydroids are water conducting cells. The term hydroid was first used by Potone in 1883 to describe the trachere elements of vascular plants and has been widely used to designate the water conducting cells of mosses. Hydroids are located centrally in the gametophytic axis and comprise what is known as the central strand. Collectively, the central strand of hydroids is termed as hydrome. In this diagram, you can very well see a transverse section through the hydrome in the rhizome of Polytrichum commune. These two cells this one cell and the other cell, they represent hydromes. These are the ultrastructures of hydromes, whereas the other diagram is of transverse section through leptoid in polytrichum commune. Now, studies of bryophytic conducting tissue in the past decade have revealed a number of similarities in the ultrastructure and physiology between leptoids and sieve elements and between hydroids and tracheids. One of such comparison is shown here in this diagram. If we could observe the structure of a hydroid and a tracheid, they share a similar outline. The tracheids, in case of equisetum, they have certain pores, but they both have a pointed end. Because of these similarities, the argument of parallel evolution of bryophytes from a non-vascularized ancestor is less tenable than the argument for the evolution of bryophyte from a vascularized ancestor. This unrevealed collection of similarities should be considered ancestral, just as a cuticle and stomata in mosses are considered persistent ancestral features. If the above statement is valid, then the conducting cells of this vascularized ancestor could be postulated to be at the structural level of hydroids and leptoids. Regressive evolution of bryophytic conducting tissue from a vascularized ancestor possessing tracheid is also probable. We have also gone through many of the changes which are required in this development of leptoid and hydroid. These leptoid and hydroids, they develop into at a later stage into xylem and phloem. These developmental details of hydroids, xylem and phloem can be seen from the previous slides, where we can see in this diagram that the hydroids are placed at the center, which will conduct water and which will gradually evolve into xylem, and the leptoids will gradually evolve into phloem, and it will conduct nutrient material in both the directions. So, as we have seen that these features of evolution of various features such as vasculature or alternation of generation, or you may say the various adaptive features, are those characteristics which helped the movement of plants from an aquatic habitat towards terrestrial habitat. In our next class, we will or in our next lecture, we will see various other features such as seed development or evolution of seed habitat. In case of bryophytes, if we could see, the seed habit was for the first time started setting with the formation of embryos. Embryos encased in a protective structure gave rise to a future fruit formation. These protective structures around an embryo ensured that the dispersal and survival chances of these embryos are much more as compared to their ancestral aquatic plants. These development of seed habit took place gradually from the gametophytes of bryophytes. Amongst the bryophytes also, if we see the Hepaticopsida, we will find that the spores are single celled and contained inside one complete sporophytic chamber. Gradually, when we move towards more evolved bryophytes such as mosses, we will find unique structures or specialized structures which bear these spores, and they have developed a unique mechanism to dispersal of these spores. Now, these seed habits gradually evolved from bryophytes towards pteridophytes. In pteridophytes, we will see these spores have more specialized structures. Now, all these bryophytic and pteridophytic spores, they are gametophytic or haploidic in nature. As we move from pteridophytes towards gymnosperm, we will start finding out two seeds in the form of a protective cover inside which the embryo is situated, and then gradually the seed habit is more established in angiosperms, where we will find that the reproductive structure, that is flower at its base in the ovary, possesses seed after the fertilization of male and female gametes. So, in our further lectures to come, we will see various such features and common features of various Archegoniate members. Thank you so much.